Okay, let's unpack this. We are taking a deep dive into something truly fascinating today. How researchers actually measure the unseen, the inner workings of the animal mind, turning these internal cognitive states into concrete, measurable behaviors.
It really is the ultimate exercise in scientific translation, isn't it? We're trying to take these abstract human mental concepts,
Hmm.
things like planning your meals for next week or experiencing a really specific memory from high school, and then designing behavioral tests that are so precise they can capture that exact cognitive capacity in, well, everything from a rat to a raven.
Exactly. And our source material today, it's kind of split into two major but related fields. First, we've got this deep biomedical work. It's focused on developing really rigorous animal models for specific human cognitive impairments, particularly looking at working memory deficits you see in conditions like schizophrenia.
Right. And second, we're looking at what you might call the frontier of complex high-level cognition. So designing tests for things like episodic-like memory, mental time travel, and trying to figure out what constitutes true future planning across really different species, primates, birds, even rodents.
So our mission here is to uncover those specific behavioral paradigms, from the highly automated operant boxes they use for like high throughput drug screening, all the
Mhm.
way to really creative maze designs. Basically how scientists objectively measure these complex concepts.
And what's really fascinating, I think, is that by carefully studying how animals remember the past and anticipate the future, well, we're systematically challenging these deep-seated traditional assumptions about which species actually possess sophisticated intelligence.
Mhm.
Get ready to have some of your ideas about brain evolution possibly flipped on their head.
Okay, let's start with segment one, working memory, or WM. This is where the scientific pressure is, well, it's immense, because the research is deeply translational, aimed right at drug discovery for some really devastating conditions. The core challenge here seems to be understanding the nature of that WM impairment in human patients.
That's exactly right. When you test human patients with schizophrenia, their working memory is reliably impaired, no question. But here's the crucial nuance. This impairment is not typically delay dependent, at least not once the delay gets longer than about 1 second.
Wait, hang on. I need to pause on that. That sounds really counterintuitive. I mean, if you test my memory, the longer the delay, the worse my performance is going to be, right? That just seems natural. But you're saying in these patients, the deficit isn't really about the delay duration itself.
Correct, largely. It means that the standard animal memory tests, where performance pretty much universally degrades the longer you make the delay, well, they might not accurately model the specific deficit researchers are targeting in humans, so they need specialized tasks. Tasks focused not just on duration, but on two other key things, maintaining a specific goal in mind and interference control.
Right. So to test that goal maintenance piece, what's the paradigm that's become the kind of gold standard in the translational field?
They've really converged on the delayed non-matching to position paradigm, DNMTP for short, and often using the operant version. You can think of it as a very structured game for a rat.
Okay, tell us how it works. Lay it out.
So the rat is placed in an operant chamber, a special box. First, it samples one lever, that's the sample phase. The lever might be on the left or maybe the right. Then, after a very precisely controlled delay, could be short, could be long, the rat is presented with two levers, both left and right. And here's the key. It has to choose the non-matching position to get the reward.
Ah, okay. So if it's sampled the left lever initially, it has to remember that, but then choose the right one after the delay, remembering the original spot and then kind of overriding that to pick the new one. And why is the operant box version the preferred method here?
Mostly because of scale and control. In a drug discovery environment, you need consistency, you need high throughput capability, and you need easy blinding so the experimenters don't know which rats got which drug. The operant version, using these standardized boxes and automated timing, it just provides all of that. It allows for rapid screening of new potential drugs.
Okay, moving to the neurobiology. The sources seem pretty clear that the prefrontal cortex, the PFC, is necessary for success on the DNMTP task. But the role of the hippocampus, the HPC, that seems more debated. Why the ambiguity there?
Yeah, it seems to be highly dependent on the specific behavioral strategy the animal uses. The hippocampus, as you know, is very involved in spatial memory. So if the task isn't designed quite right, the rat might use what they call mediating strategies. Things like just staying physically near the sample lever location during the delay. That kind of reduces the need for pure working memory and seems to minimize the HPC involvement. Researchers have found that procedures designed specifically to prevent those sorts of cheats, well, they appear to increase the necessary involvement of the HPC.
That's a really subtle but critical methodological point. Okay, now let's talk pharmacology, because this sounds like where the model sometimes gets tricky. When researchers tested existing antipsychotics drugs like haloperidol, risperidone, clozapine in normal, healthy rats doing this DNMTP task, they found something kind of weird, right?
They did. They observed these unexpected procognitive effects. And just to be clear, procognitive means the drug actually improved the normal rat's performance. It made them better at the task.
But hang on, these are drugs used to treat a condition that causes cognitive impairment in humans. Why on earth would they make a healthy rat smarter? That seems completely counter to their clinical use.
It's a real paradox, isn't it? It's the challenge of modeling a disease state in a healthy animal. You're trying to reverse a deficit that isn't there. So if the baseline subject is already functioning normally, the drug might just push the system into some kind of hyper-efficient state. This observation really just hammers home how difficult it is to accurately translate human drug effects using healthy animal models sometimes. They might just improve normal function rather than actually fixing an impaired function.
Okay, right. Let's pivot then to the second major component of WM needed for this translational work, interference control. So this is the capacity to protect what you're currently holding in working memory from competing stimuli, like trying to focus when three different people are talking to you at once.
Exactly. And in patients with schizophrenia, a key deficit is rejecting repeated stimuli. It's similar to what you see in humans performing the NBAC task. For rodents, researchers often use temporal order memory tasks. These are typically done in mazes or maybe on whole boards. The discrete temporal relational test is particularly interesting here.
And this is where we see another one of those counterintuitive findings, this time regarding lag, right?
Absolutely. You'd normally assume, I think, that the closer two items are in time, the easier it should be to remember them sequentially. But in this discrete temporal relational test, performance often actually improves with increasing lag. That is, the longer the time separation between the items the animal has to remember the order of.
So let me get this straight. If you ask the rat to remember the sequence of two specific things, they do better if those two things happen further apart in time than if they happened close together. Why would that be?
Well, the thinking is that what they're really testing isn't just pure order memory, but the animal's temporal discriminability. It might be easier for the brain to sort of file away memories that are clearly separated in time. That wider separation likely provides a clearer temporal context marker for each item, making the memories less likely to interfere with each other.
Ah, okay, that makes a certain kind of sense. So we've covered how they hold data in mind. Let's transition into segment 2. Memory capacity, the sheer amount of information they can maintain in WM.
Right. In humans, we know that people with damage to the hippocampus can show a reduced capacity span for things like remembering visual patterns or colors. So to model this in animals, the goal was to find a rodent test that mirrored that kind of shrinking capacity limit.
And this is where the odor span task, or OST, comes in.
Yes, this is really a beautiful example of a task that incrementally increases the cognitive demand. Rodents are trained to remember a continuously growing list of odors they've already smelled. They only get rewarded if they choose a novel odor in the sequence. If they happen to pick an odor they've smelled before in that session, they fail that trial.
So basically, you keep adding odors, testing their capacity until the list just gets too long, and the rat can no longer hold all the previous odors in its memory bank.
Precisely. And the really high relevance here is that performance on the OST seems to map directly onto the kind of capacity limits observed in schizophrenia patients. Plus, it's incredibly pharmacologically sensitive. For instance, nicotinic agonists like nicotine itself can actually increase the span capacity in rats, while muscarinic cholinergic antagonists like scopolamine significantly impair their performance.
Wow. So we have a model that measures capacity and responds to known chemical modulators of attention and memory. That's powerful. Now, let's zoom out a bit to neuroecology. If capacity is so vital, we should probably see it reflected in the physical brain hardware across different species, right? Depending on their needs.
We absolutely do. Evolution seems to provide the necessary architecture based on demand. Take food-hoarding birds, like chickadees. Their need for high memory capacity isn't constant throughout the year. It varies seasonally, and it peaks when they're storing massive amounts of food for the winter. Thousands of locations sometimes.
And what happens to their brains during this peak storage season?
Well, that peak demand correlates directly with a measurably larger hippocampus. It actually has more cells and incorporates new neurons right at the time of peak storage. It shows this incredible neural flexibility driven purely by environmental demand.
That's amazing. And we see that comparison even between different species of kangaroo rats, which is just brilliant.
It really is a perfect natural experiment. You've got the scatter-hoarding Merriam's kangaroo rats.
Hmm.
These guys hide food widely in maybe thousands of tiny little caches. And then you compare them to the bannertail kangaroo rats, who
Hmm.
are larder hoarders. They keep pretty much all their food in one single central spot, like a pantry.
So one is basically managing a massive distributed database, while the other is just managing a single file location.
Exactly put. And sure enough, the scatter-hoarding Merriams have a significantly larger hippocampus. The higher reliance on remembering all those scattered caches demands bigger memory hardware. Simple as that.
Okay, fantastic. We've covered memory capacity in the present. Let's jump now to segment 3, which is truly, well, it's mind-bending stuff. Mental time travel. Can animals actually recall specific past events and maybe anticipate specific future events?
This really hinges on the distinction between two fundamental types of long-term memory. We have semantic memory first. That's your factual knowledge. Paris is the capital of France. But then we have episodic memory. And these are specific personal events tied to your subjective sense of time. You remember being somewhere doing something linked to a specific moment. It's the re-experiencing part.
And the subjective experience linked to personal time. That's what Endel Tolving called autonoetic consciousness, right? That self-knowing awareness through time.
Exactly. And the foundational human evidence for this link, it comes from the very famous case study of patient Casey. He suffered severe damage to his hippocampus. And the result was he completely lost the ability to recall any personal past events. His episodic memory was gone. But critically, he retained all his factual knowledge, his semantic memory, and even his working memory was intact.
And what was the most profound insight that came from studying Casey?
It was that he was equally impaired in remembering his personal past and thinking about his personal future. He literally couldn't imagine himself doing anything specific tomorrow or next week. This strongly suggests that the capacity for mental time travel, both looking backward episodically and projecting forward into the future, is rooted in a single, unified neurocognitive mechanism. It's not like two separate filing systems. It's one system that manages past and potential future personal experiences.
Okay, so how on earth do you translate that profound insight into a test for, say, a bird? How do you possibly test a scrub jay for autonoetic consciousness?
Well, researchers came up with a really ingenious design, the what, where, when test, or WWW test, and they capitalized on the scrub jay's natural food-catching behavior.
Okay, walk us through the setup. What did they do?
Right, so they gave the scrub jays two different food options to cache. They offered preferred but highly perishable wax worms, and then less preferred but non-perishable peanuts. The birds cached both of these in different locations that the researchers knew.
Got it. Then the crucial time variable is introduced.
Yes, exactly. After a short retention interval, let's say 4 hours, the birds were allowed back. And they strongly preferred searching for those highly desirable worms they'd cached. Makes sense. But after a long interval, say 124 hours, by which time those tasty worms would have decayed and become pretty nasty, they skipped the worm caches entirely. They preferentially searched for the non-perishable peanuts instead.
Wow. So they remembered the what, which food type, the where, which location, and crucially, when the passage of time coupled with their knowledge of perishability. They were essentially anticipating the future state of the food based on their memory of when they stored it.
It's incredibly compelling evidence for what we call episodic-like memory in animals, but, critics rightly argued, well, maybe they're just following a learned rule, like a semantic memory rule. If a long time has passed, ignore the worm spots.
Right, that's a fair point. So that's why the unexpected question test was developed, to really try and nail down if the animal is retrieving a spontaneous incidental memory rather than just executing a learned response.
Precisely. The absolute key is forcing the animal to recall an event it hadn't specifically prepared to remember later. Researchers use pigeons for this. They train them on specific actions like peck a yellow light or maybe refrain from pecking a blue light. And then crucially, they gave them a mechanism using different symbols to answer a question about that action later.
And the genius part comes when they unexpectedly presented that question after the birds had just performed one of these incidental actions, right? They essentially asked the pigeon out of the blue, hey, what did you just do?
Exactly. And the pigeons reliably chose the comparison stimuli, the symbols, that matched their previous incidental action. They correctly answered the unexpected question. This strongly demonstrates they had encoded the details of that event episodically without having a prepared plan or intention to retrieve that specific memory later on.
That's fascinating. Okay, this leads us naturally to what feels like the peak of this cognitive capacity. Segment 4, future planning. True future planning.
Yeah, and for true future planning, The scientific criterion is very strict. It's often called the Bischoff-Kohler test or hypothesis. The core idea is the action must be taken in the complete absence of a current need or drive state, purely in anticipation of a future need.
That's the famous spoon test analogy you sometimes hear, isn't it? Like, you pack a swimsuit in the middle of winter because you anticipate needing it on a future beach vacation, even though you have absolutely zero immediate need for a swimsuit right then.
Exactly that. And most primates, surprisingly, really struggle with the strictest version of this test. Monkeys, for example, often exhibit what's called temporal myopia. Basically, they seem indifferent to getting a much larger food reward later if a smaller immediate reward satisfies their hunger right now. They find it hard to sacrifice immediate gratification for a bigger long-term gain.
However, chimpanzees have demonstrated real planning capacity, haven't they? Like collecting the right tool in one place, carrying it around for hours sometimes, and then using it much later in a totally different location. That clearly suggests planning the action far ahead of the actual need.
Absolutely. But what is truly revolutionary, and what our sources really emphasize, is that ravens are now challenging great apes in this domain of future planning. Researchers cleverly adapted the primate cognition test battery, the PCTB, into the corvid cognition test battery, or CCTB. This allowed for really rigorous systematic comparisons across these very different species.
And the corvids, specifically scrub jays in some studies, actually passed the Bischoff-Kohler criterion.
They did, quite convincingly. In one key study, scrub jays demonstrated planning by caching the specific type of food they knew from prior experience they would not receive in their overnight compartment the next morning. They were acting entirely in the absence of a current need. They weren't hungry at the time of caching purely to prevent anticipated future hunger based on what they expected to be deprived of later.
That's incredible. But the quantitative results from the broader CCTB comparing ravens to apes are what really stopped me in my tracks when reading this.
Yeah, the data is pretty stunning. The research showed that ravens exhibited these full-blown cognitive skills across a range of tasks by just 4 months of age, indicating incredibly rapid development. And quantitatively, their overall performance in tasks measuring physical cognition, things like understanding quantities and causality, and also social cognition like communication and precursors to theory of mind. Their performance was found to be comparable, statistically indistinguishable in many cases, to that of adult chimpanzees and orangutans.
Ravens.
Hmm.
Comparable to adult great apes. And achieving this by 4 months old. Let that sink in.
It's profound, especially for our understanding of neurobiology. This level of advanced cognition really challenges the long-held idea that cognitive sophistication is strictly dependent on the mammalian neocortical architecture. Passerine brains, like those of ravens, have evolved a completely different structure, the avian pallium. But get this, they can pack in sometimes twice as many neurons as a primate brain of the exact same size. They've clearly achieved complex intelligence via a totally alternate evolutionary route.
Wow. Okay, so we've covered a huge range here. Everything from the technical DNMTP and odor span tasks, showing how researchers model subtle cognitive deficits in rodents for drug screening, all the way to the what, where, when test and the Bischoff-Kohler hypothesis, which are probing the absolute limits of animal consciousness and their perception of time.
We basically established that we have birds planning for future hunger, we have rats holding complex high-demand sequences in their working memory, and crucially, all of this is being measured through meticulously designed objective behavioral paradigms.
So what does this all mean then? For the bigger picture of intelligence itself, where does this leave us?
Well, the sources clearly show ravens achieving cognitive parity with great apes on many complex tasks, despite having evolved brain structures that are fundamentally different from mammals. And this raises a really important, maybe provocative question for you, the listener, to think about. If sophisticated cognitive abilities like future planning and episodic-like recall can apparently be developed by such incredibly disparate neural systems, the avian pallium versus the primate neocortex, should we maybe abandon the idea that intelligence is intrinsically tied to a single specific mammalian evolutionary blueprint? And perhaps instead, should we focus more on figuring out what universal functional principles might govern the emergence of complex cognition wherever it appears across the entire tree of life?